Hello again, I'm Dr. Cary Harding.
And I'm Dr. Daelyn Richards.
And we are very excited to share with you our progress towards finding a cure for PKU using gene therapy.
Now I'm going to discuss our research. PKU gene therapy is actually quite simple. We take our therapeutic DNA, package it into a small virus called adeno-associated virus, or AAV, and it does what it's evolved over millions of years to do, and that's deliver the DNA to the target cells of our patients. And for PKU, that's the liver, where we want to restore enzyme activity. There are several types of PKU gene therapy, and you can think of them kind of like tattoos. On one hand, you can get temporary tattoos, which these days can look quite convincingly real and stay on for several months at a time. But because they are not embedded within the body, they fade away and disappear over time. That is in contrast to permanent ink tattoos. These tattoos embed ink directly into the body, so they stay on for life. These types of tattoos are kind of like gene therapy. On one side, you have gene addition therapy. This type of gene therapy is dependent on the stability of liver cells because it is not embedded in the genome. You can think of it like free-floating DNA within the liver cells, and as cells divide, the therapy is lost. As you can imagine, this approach would not be conducive in children whose livers are constantly dividing and growing. In fact, when we treat infant mice with this therapy, it's lost in a matter of days. The other type of gene therapy is called gene editing. This type of gene therapy actually goes into the genome and restores DNA function. As cells divide, the genome and the restored DNA within it are retained throughout life. The other piece of information that is important to keep in mind as we discuss gene therapy is that it can only be administered once. We have one shot to make it right. So our goal in developing gene editing therapy is to make sure it is curative for life before we bring it into the clinic. Our lab's involvement in gene editing research started when I entered the Harding lab as a graduate student in 2014. My goal was to address the temporary nature of gene addition therapy by pursuing gene editing with the aim of performing the first ever permanent gene therapy in PKU mice from birth. To accomplish this, we harnessed the revolutionary CRISPR-Cas9 system, which essentially is a way to perform microsurgery on the genome. The Cas9 enzyme is like a scalpel, and we tell it exactly where to slice the DNA. Then, as a part of this therapy, we provide a repair template of DNA that the cell can incorporate into the genome, which will restore enzymatic function. One hurdle we had to address was that this system is quite large, but our virus is very small. So we employed a dual AAV system in which one viral particle carries the Cas9, or the microscopic scalpel, if you will, and the other viral particle carries the repair template DNA that restores enzymatic function. What we do is mix these viral particles together and inject them into mouse pups within a day or so of being born when they look like this. Then the viruses carry the gene editing components to the liver cells where they perform microsurgery on the genome, restoring enzymatic function in the liver, which can then act to metabolize excess phenylalanine in the diet. When they grow up, they look more like this. Now, when you look at the mouse in the middle, this is an untreated PKU mouse. You can see its coat color is light brown or hypopigmented, as compared to non-PKU animals that are black. And this is because of high levels of Phe inhibiting proper melanin production, which is the molecule that gives our skin and hair pigment. All of our gene-edited PKU animals were indistinguishable from their non-PKU litter mates from the outside, which was really exciting to us. But the next question was, what was going on the inside at the molecular level? Here are the results of this study. All mice treated with our optimized approach were downgraded from classical PKU, having blood Phe levels around 35 on an unrestricted diet, down to mild PKU, with blood Phe levels less than 15 and even some less than 6. While this isn't fully curative, it is certainly a step in the right direction and an area of improvement that needs further optimization. Excitingly, we found that this therapy was stable throughout life and even multiple pregnancies, and that this therapy had ameliorated the effects of maternal PKU syndrome. Ultimately, we found that this therapy was, in fact, permanent. Now, there are a few areas we need to improve. Obviously, we need to get the levels down to 1, but we also need to address the design. The mice in this initial study had a single point mutation in the DNA that caused PKU, so this experiment only needed to correct that single molecule of DNA. But in the human population, there are over 1,000 mutations in this gene that cause PKU. So this approach wouldn't be feasible to translate into the clinic. We need to think bigger, like full gene-sized bigger, and that's exactly what I'm doing now as I continue gene editing research in the Harding Laboratory as a medical student. We designed a new approach that still uses the Cas9 system to assist with the microsurgery on the genome, but now we're inserting an entire mini gene cassette, making this new approach broadly applicable to all humans with PKU. So far, I've treated 3 PKU mice with this therapy, and I'm excited to say that we're seeing results. Just as in the last study, all of these mice treated have been downgraded from classical to mild PKU, with blood Phe levels less than 15 on an unrestricted diet. And that this therapy provides stable, lifelong correction in any PKU genotype. Now I still need to finish this work, and moving forward, we need to find a way to increase the restoration of enzyme activity to cure PKU from birth, so that one day we can allow children born with PKU a life without limits on what they can eat, or without fear of hurting their brain. The research we do in this lab requires a lot of resources and hard work. For a single gene therapy experiment, we spend countless hours meticulously designing our therapeutic approach. Then we synthesize the DNA in a special lab, amplify it, and then send it off to be packaged into the AAV virus. We perform all of the procedures on the mice and have veterinarians help us care for them so they are safe and well taken care of. And then we run a plethora of experiments to analyze the gene therapy at the molecular level. The cost of performing this type of cutting-edge research in our lab for 1 year is roughly $300,000. We know the PKU community deserves better. So please help us, because this research doesn't happen without you. We still have a lot of work to do to find a cure for PKU.
And none of our work would have been possible without the generous support of donors like yourselves and the National PKU Alliance.
So if you're committed to sustaining our research efforts, please raise your paddles, lift the limits, and fund a cure.